It is a pleasure to be here because this is the epicenter of ergothioneine research, as you might have noticed in Barry's wonderful presentation. So this is the question I have. Is ergothioneine a longevity vitamin, like Barry mentioned, that can help to unlock healthy aging? Well, in the next 30 minutes or so, I hope to give some answers to that question. But spoiler alert, it has something to do with the way we grow our food, as you might see there. So as Barry mentioned, it was discovered in 1909. It is a small molecule amino acid, colorless, odorless, water-soluble, and but it's very stable antioxidant because of that. It's in the thione form at physiological pH rather than the sulfhydryl form, which would make it less stable. So in the 1950s, some researchers in the United States discovered that various animals and even humans had high levels of ergothioneine in their blood, like millimolar levels, and we're saying, wow, what's it doing there? They were able to trace it back to the food or the feed that they were getting, and but nothing happened after that, like Barry mentioned. It's not produced by us or animals or plants. It's only produced by fungi, blue-green algae, and a few bacteria. And it's concentrated in mushrooms, and that's how I got involved. I was doing mushroom research back years ago, and in about 2004, one of my colleagues on campus, who was a biochemist, said, hey, Bob, you should look into whether mushrooms have ergothioneine. I think they probably do. So we did that, and sure enough, it's very high concentration in mushrooms because it's just a big ball of fungus. So like Barry mentioned, in 2005, the ergothioneine transporter was discovered by Dirk Grundemann at the University of Cologne, and when everybody saw that, much interest came up, including us. As he mentioned, it's highly expressed in many tissues. It's a potent antioxidant, but also anti-inflammatory. And as Barry mentioned, it declines with age, but it declines faster in people with chronic diseases. So in 2010, Solomon Snyder, a famous neuroscientist at Johns Hopkins University, published a paper in Cell Death and Differentiation and about ergothioneine. It's the only paper he ever published on it. Unfortunately, he retired shortly thereafter. But he showed this slide, which is a diagram of the cell, and you can see the ergothioneine transporter is in the membrane and transports it across the membrane. And Dr. Grundemann calls it ETT now rather than OCT1, so that's why it's listed here. So it gets into the cell, and then it gets into the mitochondria, like Barry mentioned. And as you know, all the metabolism going on there's a lot of free radicals produced, a lot of reactive oxygen species, which occur in the mitochondria and damage it, but ergothioneine tends to block it. Some of they leak out into the cell or outside the mitochondria, and all of these contribute to protein oxidation, lipid peroxidation, and glutathione oxidation, the major antioxidant. It also protects from UV radiation and from DNA damage due to these free radicals. I think he was speculating a lot on that, but now we know that he knew what he was talking about. Well, so I was working with mushrooms, and the people that was supporting me at the time said, well, is it bioavailable? You know, we don't know that. So we did a study in the Dr. Pennycrest Etherton's lab in the nutrition department, where we fed a mashed potatoes and gravy meal to students, and their blood was taken at 0, 2, 4, and 6 hours, and we measured the difference between 0 time and those times, and you could see that within an hour it's into the blood. This was whole blood. We didn't have the ability to measure it in plasma at the time. This amount of mushroom powder equated to about 3 to 4 milligrams of ergothioneine. So later I got involved in a study at Mount Sinai Medical School, where there was a number of people attending a clinic there that were given 100 grams of mushrooms a day for 16 weeks. They were given meals that had prepared by a chef that had 100 grams of white button mushrooms a day. And we then had the plasma. We measured the ergothioneine level. Oops. As you can see, it basically doubles during the 16 weeks, and then after they go off, it drops off. If you look at the AGEs in the blood, which are advanced glycation end products, very pro-inflammatory, you get just the opposite effect, which is a good thing. Ergothioneine in the blood is reducing those. A little bit of effect on the ORAC value, and interestingly enough, adiponectin, which is an antioxidant hormone, increased and didn't drop off yet after they went off. Well, my colleague at our medical school in Hershey, Dr. Jibril Bah, has published like 4 epidemiological studies in the past few years, like this one, and showing that mushroom consumption had an effect, positive effect on depression and cancer and cognition and so on and so forth. But the problem is, like everything I've shown you so far, we're talking about mushrooms, not ergothioneine. It does have ergothioneine, but it was basically done with mushrooms. And as you know, there's other things in there that could be affecting it. Okay, so time came about the time I retired 15 years ago, and I got to thinking, there's half of the people in the United States don't eat mushrooms, or very few. So where are they getting it? And so my hypothesis was they must be getting it from, they must be getting it from the soil. And there are fungi in the soil which produce it, like with mushrooms. You have the mycelium in the soil or in the substrate. It's passed to the mushrooms, and this shows that it's very high level that goes to the humans. In green plants, they're photosynthesizing. They're taking in carbon dioxide, giving off oxygen, and they're sending chemicals down to the roots down here. And there are fungi associated with the roots called arbuscular mycorrhizal fungi, which have an intimate relationship with roots. They can't, these fungi can't grow only in the presence of the roots. There's a symbiotic relationship there. So the photosynthase that are coming down are feeding the mycelium down here, and on the other hand, the mycelium is passing up water and minerals and ergothioneine to the plant. And of course, then animals eat the plants, it comes to us one way or the other. So we have a dynamic relationship here that is important, as you'll point out. I'll try to point out to you later. Well, talking about mushrooms, we did this study a number of years ago, and it shows that there's quite a variation. Ergothioneine in the light bars, you see lowest in the white button, and then goes up. Yellow oyster is high, and then porcini, which is a wild mushroom, is the highest, but unfortunately they can't be cultivated. But the Italians, they go out and hunt them. And but we also did glutathione, because one of the co-authors in this paper was a glutathione expert, and he mentioned that this is the highest level of any food that he's ever seen as far as glutathione is concerned. So we have a mushroom that's a high level of both of these important antioxidants. Well, this just shows levels in the normal diet. And you can see these are before we did this work, these were the highest level foods, chicken liver being the highest, and white button and gray oyster much, much higher. But somebody measured it in tempeh that was quite high in that. And of course, this is a mold fermented food. It looks something like this. You take soybeans and dehull them, boil them in water, cool them, drain them, inoculate them with a mold, Rhizopus silicosporus, incubate them for a day at warm temperatures, and you get it completely impregnated with the mycelium, the white material. And so basically, a 100-gram slice of that would have almost 10 milligrams of ergo, which is a very good source. Well, back in about 2018, there was a paper published by a bunch of French workers where they estimated the amount of ergothioneine in people from 5 different countries, the US and 4 European countries. And I took their data and calculated what it would be on the average weight person in milligrams per day. As you can see, unfortunately, Americans have the lowest, and then in this case, the Italians had the highest. So then the question is, does this have any importance in health, especially longevity? So we plotted these estimated ergothioneine consumption per day against life expectancy. And at this time, this paper was published here, and Japan wasn't part of it, but I estimated the consumption in Japan and added that as a red dot. And as you can see, there's a very nice positive association between estimated ergo consumption and life expectancy. But as you know, it's not, this is a correlation, it's not causation. But it makes you wonder. We also looked at Parkinson's disease rates, and you see that the higher the level of estimated ergothioneine, had the lower levels of this disease, and the same thing with Parkinson's disease. The other one was Parkinson's, and this is Alzheimer's. Again, this doesn't prove anything. In fact, the first time I showed this to my colleagues, they laughed at me. And, but they're not laughing anymore, I don't know. Okay, the Japanese appear to have very high life expectancy. They have low levels of Parkinson's and Alzheimer's. And I estimated they're consuming a lot of ergo. And where is it coming from? Probably, this just shows you the level in oat bran, which is a high level, but miso, soy sauce, and koji are all part of their diet, and they eat a lot of shiitake mushrooms. And shiitake mushrooms have 3 times as much ergo as white button mushrooms, and they eat 3 times as many mushrooms as we do in the United States. So this is part of the reason. Well, Barry mentioned Dr. Bruce Ames. I met him at a conference back in 2012, and he talked about ergothioneine and other things being longevity vitamins. And so I invited him to come to Penn State and give a talk, and this is just a quote from him that he gave that day. And he published this paper in the Proceeding of the National Academy of Sciences declaring these micronutrients, including ergothioneine, as longevity vitamins, and he based it on his triage theory. You all know what a triage is. It's a French word. It was first used in the First World War when there'd be mass casualty situations. The people that had the most serious injuries that were savable would get first priority to any equipment, any personnel to save them. And then the people that could be let go for a while, they would get those materials last. And if there's low levels of those supplies, they don't get any. Well, the same thing he says with ergothioneine and other with longevity vitamins. If long periods of time go when there's not enough ergothioneine in the blood, these diseases or these conditions which cause chronic disease would be allowed to occur and accumulate over time and reduce life expectancy. So this just shows the effect of dietary ergo, and Barry mentioned that 810, in his case, nanomole in the plasma, was the cutoff point. So if you had lower levels, it would be considered insufficient. And so there'd be a lot of cases where there wouldn't be enough present to prevent the oxidation of proteins and whatever and DNA damage, and you lead to severe oxidative or inflammatory disease. But if you have enough, especially the high levels, it prevents it. So that's kind of the triage theory in a thumbshell. Well, there was a number of metabolomic studies that were done recently. This one I thought was fantastic. It's done in Sweden. They were looking at 112 metabolites measured in over 3,000 people. They were all eating a healthy diet. So they wanted to find out what metabolites in their blood might be responsible for the fact that they don't get cardiac disease and they have a longer lifespan. So out of the 112 metabolites they measured, the only one that had the really high positive association with reduced coronary disease and increased lifespan was ergothioneine. And I don't think they were really expecting that. And of course, it happened. A similar study done in Japan, where they looked at elderly Japanese over a period of time that some of them got dementia, frailty, and cognitive impairment, and they measured the metabolites that were present either higher or lower than in the healthy people, and they drew a circle around all the ones that are involved in dementia, 33 compounds, frailty, 15 compounds, cognitive impairment, 6 compounds, and you might notice there's an interesting thing here where all of them occur together, and there's 2 metabolites, ergothioneine and tryptophan. And of course, tryptophan is the precursor of serotonin, so maybe that's. Well, the Japanese, again, Barry mentioned this study, so due to time, I'm going to move on. Okay, there's a couple of preclinical studies that one I got involved in with the medical school at Vanderbilt University. And this gentleman, Wellington Pham, is a radiological chemist. I gave a talk there a few years ago, and we got very excited. He said, I can make a radioligand of that, and we can use it to follow ergothioneine moving into. And so he could inject it in the tail of the mouse, and within a few minutes, it's going into all the different organs in the brain and everything. And after a period of time, they sacrifice the animals, and they look at the brain. And this is the Alzheimer model mouse, and this is the wild type. And you can see this is the brain. Here in the red is the eye, again the brain, and it shows that the Alzheimer model mouse has much higher levels of oxidative stress, as shown by this with the probe. Unfortunately, he wasn't able to follow up too much on that. They did a longitudinal feeding study where they treated the Alzheimer model mouse with ergo, reduced amyloid plaques, oxidative stress, and reduced glucose metabolism, and they came to the conclusion that it has the potential to prevent Alzheimer's disease, but unfortunately, they've never been able to follow up on this. They haven't been able to get support to do it. Well, another clinical model, or the clinical study, was done recently in Japan, where they fed mice ergo in their water, and then they had controls that didn't get any ergo. It was a blinded test. The mice didn't care whether they didn't know whether they were getting it or not. You get that? It's a joke. Okay. Blind mice and whatever. Anyway, they found that survival curves look like this. This is the mice that were getting just water, and these are the ones that were getting ergothioneine. The longevity increased from about 700 days to 1,000. So again, just another preclinical model. Okay, this is the study that Barry just mentioned, the pilot study. So in view of time, I'm going to pass on to that. There was another clinical study that was done in Australia using ergothioneine produced by precision fermentation, where 147 adults between 55 and 79 with memory complaints but no dementia were given 10 and 25 milligrams of ergo or placebo for 16 weeks. The plasma ergo levels did increase dose-dependent fashion. There was a subjective prospective memory and sleep initiation improved with the dose that was significant at 25 milligrams. Reaction time within groups was significant at 25. Telomere length increased in females. Okay, now we get to the bottom of the situation. This is healthy soil. Okay, it's dark because it has a lot of organic matter. There's lots of microorganisms in there. In fact, it's been estimated that a teaspoon of healthy soil contains more microorganisms than there are people on the planet. That's interesting, isn't it? And when there's a lot of ergothioneine in there, the plants can pick it up, and the food that's derived from it has high ergothioneine content. However, many of our soils are completely depleted. They don't look anything like this because they've been abused by agricultural practices that are only involved in trying to improve the yield of the crop year after year after year. Actually, Leonardo da Vinci he quoted in 1580, he said, "We know more about the movement of celestial objects than we do the soil under our feet." And I think that's more true now than it was then. So we got the idea to look at tillage, because that's where you disrupt the soil, and there's a chance of disrupting the fungi in the soil. And we have plots at Penn State where they've been going on since 1978, and no-till had the most ergothioneine in these three crops successive years, about a 30% change from no-till to heavy tillage. Interestingly enough, we also followed up with that with a group from our USDA, where they did the same thing with soybeans and wheat, but they took root samples during the crop cycle, and they measured the amount of fungi associated with the roots. And as that increased, the ergothioneine level in wheat increased. And it's just a model of what a arbuscular microbiological fungi look like. They actually impregnate the root, and you can see the cells of the root. So there's a big movement in the States to improve upon the way we grow our food to what they call regenerative agriculture, which is sort of the antithesis of conventional agriculture, where it involves minimal tillage, use of cover crops, never leaving the soil bare, multiple crop rotations, reduced pesticides and chemicals, fertilizers, and sometimes involves grazing with animals. These are just the opposite of what conventional agricultural practices are. And when they practice these, you get more fungi, more ergo, reduced inputs, so it's cheaper. You don't have all the chemical costs, the plowing and whatever. Improves the soil health and improves the environment. So a few years ago, I wrote this paper for a magazine in the food industry, Food Technology Magazine, trying to promote the idea of healthy soil, healthy people to the food industry. It was what they called a dialogue section. It was supposed to create dialogue. And what I showed was what I was telling you about the importance of regenerative methods to increase soil health, and then you get more micronutrients like ergothioneine in the food. And of course, we have this chronic disease epidemic in the United States, and I think that it may be partially responsible to the fact that we have low micronutrients in most of our food supply. Guess how many people responded to that? None, within 2 years. So it kind of shows the mentality of what's going on in the United States. So I've been retired 15 years. I don't have a lab, I don't have any students, I don't have any money. So I rely on collaborations. And these are the collaborators that I'm involved with at Penn State year in and year out. Michael, Al Phillips, Stuart Diker, who's a soil scientist, plant scientist, John Ritchie and Zachary Bitzer at our medical school in Hershey. Unfortunately, John died a couple of years ago, and Zach took his place. Also, lots of other people at our department, the College of Medicine, the Rodale Institute, Vanderbilt, USDA, ARS, and my good friends Barry and Irwin Shea, which have been very encouraging to me over the years. So that's my story, and I'm sticking to it.